Thank you so much for such a nice description about me. And I'm not 30 anymore. I'm 35 just recently. Um, yeah, so it's been a pleasure and to be here and to give, organize this neuro seminar and have the opportunity to share some knowledge and share some discovery that we have recently in Thailand. And today, what I'm going to be talking is this novel model-based approach that we use with EEG. And we started to apply this in people of different age and different population. And particularly today, we're going to talk about MCI or mild cognitive impairment. And just give you a spoiler, right? What we're going to be talking today is how we can take measurement like EEG and then use some computational modeling like machine learning. Here we can use invert encoding model to reconstruct what people attention would look like, and then try to use that to study how attention might change as we age, and how, what type of mechanism that go wrong, in the aging society and people with mild cognitive impairment. And before I start the talk, I would like to give a special thanks for these important people here in my life, in my lab. One is Panchali, and second is Kanyarat, who's devote their life to collecting data and do all the analysis and working with us for 2-3 years now. And my beloved partner, not like that, is Dr. Shaipat, who work here at Chulalongkorn, and we work very closely in collecting data across all age groups and MCI as well. And I would like to give special thanks to my student at KMUTT, Bonus Punch, and another special partner, Dr. Kanda Lerdarak, and Nong Nam King, who is in Finland right now, who collect all this data and help us establish, the database in Thailand. And I see so much potential coming out of this, and I would like to thank Rick and your team that help organize this and be our research partner in order to do this frontier neuroscience research that actually produce real impact to the society, I think. And now our lab in the Neuroscience Center for Research Innovation is located at Bangmot, KMUTT, on the 5th floor. So if you guys are interested in, partnering with us, just give me emails and you can welcome to visit anytime. First, I would like to introduce you with the mission of the center. One of the main things that we're doing is to do research and produce innovation that help improve learning, cognitive health, and well-being across the human lifespan. So what we're trying to do is to, in this study, what are the neural mechanism of different type of cognitive function and memory, and how to see how they change across age, from very young age to, older people. And then what we could do is use this as the baseline, fundamental knowledge to study other neurological disorders and neurodegenerative disease. And we use this framework that I actually learned from the Ministry of Public Health, the 3 or 4P framework. So the strategy that we're doing is first to do research that combine neural measurement like EEG, MRI, and AI in order to predict, whether a person might have a certain kind of symptom like MCI or ADHD or something like that, so that we can provide, new treatments. It could be invasive or non-invasive, and then find preventative plans that help them out. It could be environmental, it could be some exercise that they can do either cognitively or physically, right? And later on, all the knowledge that we got, that could help promote policy, for like learning or aging society. And today I'm going to be talking about aging society. And for this particular talk, I'm gonna focus on the prediction part, right? Can we catch cognitive deficit before it's too late, right? And if we can do this, this is really helpful because we can provide early intervention, preventative plans, and personalized treatments. Well, there's no animation here, but if you look at this figure on the x-axis here is years, and on the y-axis is cognitive function. As you may expect, when people age, our cognitive function across multiple domains will decline, right? And if it's declined, to the certain point, we will call that dementia, right? But what we're trying to do here is, can we use markers like EEG and MRI in order to detect whether people gonna turn into different kind of dementia, including Alzheimer? And the stage at which we think we should target right now is MCI, which is a stage between healthy aging and dementia. And from past literature, we know that MCI is the multiple cognitive domain disruption. And that includes different kind of domain. Memory, language, attention, executive function. But today, I'm gonna be focusing on just attention. And this is why. I guess many people this morning got into this traffic in front of Central World, right? Driving here or just like walking here requires so much attention, right? In order to navigate through a complex environment, you need to be able to focus on a certain kind of thing and suppress, all kind of distractors so that you can do the task at hand, right? And the ability for human being to prioritize information in our visual scene is really important, right? And here is how attention kind of work. We have very complex visual system that composed of different small areas that encode different kind of information in our visual field. It could be lines, color, cars moving to one way or another, or something like that. And these are encoded in very complex, areas in the visual system. And we have this frontal-parietal network that send those top-down signal and telling us where to look at, what feature to look at, so that you can extract the most important information, right? And that is not just sensory processing. Attention help modulate other kind of cognitive system, including memory. And studies have been proposed that attention not only modulate long-term memory, but also what having in your working memory, what information that you're holding to make good use of it, like you want to calculate some math equation, something like that, right? And this involves not only brain areas in the frontal parietal cortex, but the hippocampus, which is the place where the memory is being encoded or retrieved. And as you can see here, MOCA, Ajahn Chaipat already presented this, is the standard neurological assessment that assess all this multiple domain, right? And attention is a part of it. However, when you look at this standard assessment, like attention, it's not really attention, right? What usually people, clinicians or psychologists may ask the patients or people that come into the clinics, they would ask something like this. Can you read the list of digits, right? And repeat them in forward order. So let's say if I say 21854, can you repeat? Okay, you don't have MCI, I'm kidding. But what about this harder task? Subject has to repeat them in backward order. 5 to 8. 8 to 5. Okay, you don't have MCI. So no, okay, maybe you got disrupted in other domain, right? So you have to do all this assessment that could take like 30 minutes or 15 minutes or something like that. However, you can see that this is not attention in the way that we cognitive neuroscientists work, right? Attention requires this kind of selective process, not just holding things in memory. So in this talk, I will present you 4 studies, but it's probably going to be very easy to follow because we use only one approach. The first question is, can we use model break? Can you remove this thing from here? The Zoom, sorry, because it's blocked the view here. Can we use a model-based EEG approach to track attentional modulation in human cortex? And how can we see change in those kind of representation across different cognitive demands? So if the task is hard or easy, can we track change in attention using EEG and machine learning? Second, what are the underlying neural mechanisms that change as we age? And third, can we start to track attentional deficits in people with MCI? And fourth, whether we can use that further to classify different subtypes of MCI. And for the first one, we will talk about the first question, right? Can we use the model-based EEG approach to do this? And then I cannot move. Sorry. Oh, thank you. So what the approach that we're using actually come from study in non-human primate or monkeys. So what neuroscientists who study attention like to do is doing something like this. Train the monkeys for months and years to do simple attention demanding tasks. What monkeys doing here is try to fixate at the fixation point, right? And there will be a peripheral cue come up that direct attention to a certain location in a visual field. And then there will be the probes, and this probe will be like street in Bangkok, right? So many cars of different shape, the motorcycle, passenger, people and stuff. So here you can see that it simulates the way that selective attention might work in the real world, right? You give subject a cue and then they have to discriminate a certain shape of this visual object. And to make the task harder and make sure that subjects pay really focused attention, there's surrounding distractor that could be congruent or incongruent to the shape of the target, meaning whether the shapes match or mismatch. Okay. And when you drop electrodes in the monkey's cortex, either like in the thalamus, in the visual cortex, or frontal parietal cortex, you will see the enhancement in neural firing rate. In neurons, overlap visual field, right? Overlap receptive field to the attended target location. And if you look at the coherence between the signal from different locations in the brain, you will see the increase in a low frequency coherence. So meaning that the two regions would kind of like fire together. So somehow they communicate at the lower frequency. And as Professor Larry Frank talked in the morning, you see that there's this measurement called alpha power in the EEG that track the attentional states. And here we're going to use that signal, but then we will use the machine learning and convert it to our mental representation of attention. And the people doing the same thing in human, putting electrodes on the surface of the brain, this is called ECOG, and see some selectivity across the visual and parietal cortex. So we're gonna steal this kind of paradigm and adapt it so that it's matched to what we want to do. So what we do is that we program this behavioral task where we give the arrow cue and subject has to attend to a certain location and discriminate whether the cue location is diamond or bow tie, right? And after that, they will receive feedback whether they are right or wrong. And they have to do this as correctly and fast as possible. And we manipulate the task so that there's two conditions. One is the easy condition, I would say, where the distractor of the that surround the target match in shapes, and the other one's mismatched. And this is called congruent versus incongruent. And when you look at the level of single subjects and at different Q location, you can see that there will be this EEG wave that oscillate at alpha frequency of about 10 to 12 hertz. So if you count here, right, how the EEG oscillate, you can see there's an alpha wave like 1, 2, 3, up to 11, in 1 second. And that's why they say it's 10 to 12 hertz, right? And this is called alpha frequency. But if you plot across the whole head, you see there are certain topographic distribution that's distinct from one another, depending on the location at which people are attending to. And we will use these different patterns and the machine learning that I'm proposing here to convert that, to reconstruct what people are actually attending to and how precise they're tuning to the location of attention. And just in brief, this model is called the invert encoding model that learns the relationship between the topographic pattern of alpha desynchronization and the location at which subjects are learning, and compute the weight, of that represent those relationships in different channels. And then what we could do is invert it this way and then give the new EEG data. And then from that, we can use this to predict what people might be doing on their visual field. So if they are paying attention really hard, you might see that the reconstruction is precisely tuned at the attended location. You would expect something like the blue curve here, where you have really precisely tuned alpha-based reconstruction. But if people are not attending really well, then you might see the results like this, where the reconstruction just flat. And to just remind you again, this is somewhat like our mental representation of attention that's being created by EEG. And what's nice about EEG is that we have really good temporal resolution, so we can see when this tuning function come up. So let's say if 0 here is when the arrow cue come up, right? You might see attention come up maybe 300 milliseconds right after the cue come up. Right? So here's a behavioral result. It's pretty nice. It's showing that our tasks work. So if you look at the incongruent condition, subjects just get worse at doing the tasks, right? Because sometimes they are lured to the surrounding distractor, right? So they give the wrong answer. That's why the performance drop, right? If you look at the correct RT, so the response time in the correct trials, you can also see that subjects take longer to do the tasks that's harder. Does that make sense? Here is the results of the reconstruction. What you see here is that in the incongruent condition, subjects pay, you see really nice and really well-tuned alpha-based reconstruction, right, in an incongruent condition. And if you look at the congruent condition, you can see that the attention field is actually quite broad because it's not really attentional demanding. And if you subtract between these two, you see that you see the heat map showing this attentional spotlight that's come right after the cue and sustained throughout the target period. And we can look at this in comparison to other EEG index, like what we can obtain from the frontal cortex, which is a marker of executive function, or EF, like in Thailand, people like to say EF. You can also see this frontal negative signals is higher or more negative in the incongruent condition compared to the congruent condition. But this come up way later compared to the reconstruction itself. Okay. So here you can see that the conflict between information, either target or distractor, could indeed shape the spatial scope of attention, right? And conflict resolution is due to both early change in selective attention and also executive function, or EF, like I said, right? So next, we want to see how it's changed as a function of age. So we have our team run a lot of data across different age groups from 80 to 80 years old. And what's nice about this task is that when you look at the way that we change the task, difficulty of the task, we can make sure the subject actually perform the task equally across populations. So the results that different are not due to the difficulty different. What we can measure also is reaction time. And you can see here that people who are older just respond slower in general, right? So you see here's the red one is 20 to 30. If you go up to 30 to 40, you know, people are a little bit slower, right? But as you go older, like to 40, 60, then you get even slower. And the slowing in reaction time is quite exponential. And when you look at the alpha-based reconstruction, you can see that, you know, there's a change in the selectivity of the reconstruction starting at 30 years old. So actually the selectivity of our attention dropped as we, you know, age very early, like 30 years old. And interestingly, throughout late in adulthood, like from 30 to 40 and 60 years old, you don't see much change in selectivity anymore. So meaning that if you, people who aging healthily, their selective attention is quite intact later in adulthood, right? So the question is, ทำไมใช้ไม่ได้เลื่อนข้างหน้าหน่อยลูกเลื่อนไปข้างหน้าให้พี่หน่อยอันนี้ไม่ติดนะพี่ได้แล้ว okay. So what explains this big change later in life, right? Because there's no change in selective attention. What we found is, yeah, there's change in the frontal activity. So the slowing that happened later in life are indeed due to the decline in the frontal function, right? But later in life, the selectivity, you know, that's represented by alpha band activity actually stay intact. And this is there's an opposing story for MCI actually. Because when we did this in two different populations, one is healthy aging and one is MCI, that's age and education match. First of all, you see that, you know, they perform the task quite well. That's good, but they're slower. MCI in red is slower. And what happened with the reconstruction? People with healthy aging, they have precisely tuned attention, but with MCI, their attention is gone to hell. Like this is so broad, right? And if you look across time, you see really good tuning for healthy throughout the cue and stimulus period. But with MCI, it's really broad. It looks like Mickey Mouse, like it's like a Mickey Mouse attention, right? And you look at the difference, it's sustained throughout, you know, the period at which they have to attend. So we think that MCI probably, you know, being underlined by the deficits in sustained attention, selective attention, right? Which is not impaired in healthy aging. But when you look at the frontal activity, there's no change. So this is showing that MCI might exhibit this kind of selective deficits in visual attention. Okay, and next, I'm gonna be very quick now. 5 minutes left, wow, I have time. Okay, so in the first part, which is, I think it's even more interesting. What we are doing now is that whether we can use this kind of method to define different types, right? And we're going about this in a very shitty way because we think, you know, our measure attention, right? And then Mokka has attention domain, right? So why don't we just split subject based on their attention scores, right? So we collected more data. And then when we have enough, then we split them into the one with intact attention. So from the attention domain that I showed you earlier, tell subject are being told whether they have to repeat the number in forward and backward, right? we got those scores and then divided them into the higher group or the lower groups. And we got about 17 subjects here and 13 subjects here. What we see is that when we look at hit rates or accuracy, you see that there's a significant drop in the accuracy in the incongruent trials. In the MCI with attention deficits. So if they have lower attentional performance, their performance here is also lower. And overall, their reaction time is higher, right? So if the patients exhibit more attention deficit, then in this kind of queuing task, they're even slower than the one with attention intact and the one that's healthy aging. And when we now look at the reconstruction, interesting results emerge. If you look at the healthy control, their attention is like what I showed before. It's really nice in both easy and hard tasks, right? And when you compute the difference, there's not much different whether the task is hard or easy. They always have really good attention. You see the yellow lines or the yellow blobs here? It's really good. But when you look at MCI with intact attention, what happened is that they are also good, but only good in the hard tasks. So in the tasks where they have to put the effort, then you see this nice attention. So you see that in an incongruent task where the surrounding distractor mismatched with the target, then you see this nice peak of alpha-based reconstruction. And if you do the difference here, you see that there's a significant difference very early on, even like the younger group, right? But when you look at the people with attention deficits, in general, you see a drop, in the strength of the reconstruction compared to the one with attention intact and the healthy aging. And we think this is actually showing that our EEG reconstruction measure really sensitive to attention deficits in MCI. Lastly, when we look at the frontal cortex, the results switch. So you can see that, right? In this case, the attention fields are somewhat the same, right? So somehow maybe the frontal cortex might have to engage in order to solve this conflict later in time. And we see that. So if you look at the healthy aging, you see the difference in the frontal activity in the incongruent and congruent condition. But in MCI with intact attention, as you may see, there's difference between these two already. So conflict might be mitigated already early on in selection. That's why we don't see any difference in the frontal cortex at all. But in a subject with attention deficit, right, we don't see this different in the reconstruction, and overall attention is very broad in this group. What we see is kind of enhanced, difference in the prefrontal cortex. So you can see that as we age and as our attention is being disrupted, let's say in MCI, this frontal and parietal cortex, somehow play a very distinct role, in terms of supporting attentional function that have left, as we age. And this is overall comparison between the results, right? If you look at the reconstruction in healthy aging, it's quite nice between the two. But in terms of executive function, the frontal cortex has to kick in to solve conflict. But with attention intact one, you see the attention kick in when the task is difficult enough, but the frontal cortex doesn't have to work really hard anymore, right? But in the attention deficit one, you can see that overall attention field is not as selective as it should be. That's why, the executive function might have to compensate for, this dysfunction early on in time. And this might be responsible for, the modulation that we see in behavior, both accuracy and reaction time. Yeah. So in conclusion, this study is showing that different MCI subtypes may exhibit different type of neural deficit underlying selective attention, either parietal or frontal. And this kind of proves that our model-based EEG approach could help classify different MCI subtypes based on their attentional and executive function. And the future direction that what we're aiming to do is to further look at memory, like working memory and long-term memory, and see if our, we can build useful index that look deeper in terms of severity and different type, subtypes classification methods like Ajahn Chaipat used in his unsupervised learning clustering method, right? And we can, start with the provide treatment. We are investigating the effect of non-invasive brain stimulation in combination with this EEG measure, and also looking at, and also risk support the intervention part as well, where we now conducting experiments to look at the effect of the joint effect of exercise and cognitive training, on attention function. And maybe we'll see next year the progress of this research. And lastly, I would like to thank our grant agency, from NRCT, MSPSO, SKW, the Ministry of Science and Technology changed to Ministry of Higher Education, Science, Research and Innovation, Asahi Glass Foundation, RISC, and MQDC, and all of my collaborative partners. And thank you so much to everyone.
Thank you very much, Dr. Chan, for very interesting data about MCI, and we're looking forward to see and hear more about your future research, and we understand better how we prevent the MCI.
Maybe.
Maybe. Okay, now is a Q&A question. So if anyone has any question, so because of the time, our time is running out, I like to have one question. Yes.
Yes.
Such an interesting talk. But for the first part, your measurement rely on classifying EEG based on the topography of alpha, right?
Band synchronization.
Which means you mainly focus on topography and ignore timing and frequency information in your data. Would it be possible to combine both temporal frequency and topography into the same model?
Temporal frequency, you mean like other frequency or?
Yes. So
yeah, definitely. I think actually what we observe, and is actually the other parts of what we looking at is instantaneous frequency.
Right.
What we actually observe, right, in Parkinson's group, like Chaipat presented, or actually in MCI group, we see this kind of systematic shift with slowing in alpha power, right? And we are actually investigating that. Also, we could definitely look at other frequency, like theta band oscillation or other beta band or something like that. But this is a focus right now because like alpha thing is a focus right now because we just know so much about alpha up to this point. But there's so much work that we could do. Like our team, Ajahn Kanun, is also looking at Granger causality or something like that, and see, whether like this frequency or other frequency at different location might have this kind of change in terms of the direction of the communication between the two areas, something like that, and how it's modulated by aging and MCI. Yeah. Okay. Thank you so much for the question.
Thank you.